Hello everyone, my name is Balnur. I am a PhD student in Biomedical Sciences at Nazarbayev University School of Engineering and Digital Sciences. In this lecture, I will introduce you to the area of my research, which is nanoparticles as carriers for targeted drug delivery in cancer treatment. Hopefully, after the lecture, you will have basic knowledge of cancer, current therapy that used to fight tumor, and why nanotechnology attracting more and more attention of biomedical researchers. Moreover, I will talk about my thesis project and show some preliminary data and introduce you to the PhD program at Nazarbayev University. As you know from previous lectures, cancer is a group of more than 100 diseases that involve the uncontrolled division of cells. Also, cancer can develop in any tissue of the body, and each type of cancer has its unique features. However, one defining feature of cancer is the rapid creation of abnormal cells that grow beyond their usual boundaries, which later can invade adjacent parts of the body and spread to other organs. Cancer is the main problem of humanity because it is the second leading cause of mortality in the world. Moreover, cancer affects the economy and its impact increasing each year. Other significant problem related to the cancer occurrence is inaccessible diagnostics of cancer-causing infections such as hepatitis and human papillomavirus in low and middle-income countries, and those infections are responsible for up to 25% of cancer cases. For example, cervical cancer is caused by sexually transmitted infection with certain types of HPV, and two types of HPV 16 and 18 cause 70% of cervical cancer. According to WHO, only 1 in 5 low and middle-income countries have the essential data to drive cancer policy. Thus, the late-stage presentation and inaccessible diagnosis and treatment are common in that countries. That's why approximately 70% of deaths from cancer occur in low and middle-income countries. Here I want to stress again that bad behavioral and dietary habits cause 1/3 of deaths from cancer, and it includes high body mass index, low fruit and vegetable intake, lack of physical activity, tobacco and alcohol use. Here you can see statistics taken from WHO, and the latest data available on cancer is from 2018. However, if we look across the timeline of cancer incidence and deaths globally, we can say that it's increasing significantly each year. So in 2018 there were 18.1 million new cases and 9.6 million deaths. Globally, about 1 in 6 deaths is due to cancer. Also, you can see the pie chart representation of the cancer incidence and deaths by regions. My main area of interest is breast cancer at the moment, and it is the most common cancer among women, affecting 2.1 million women, and also it causes the greatest number of cancer-related deaths among women. In 2018, it is estimated that 627,000 women died from breast cancer. That is approximately 15% of all cancer deaths among women. In recent years, doctors are increasingly using genetics information about breast cancer cells in order to categorize breast cancer. These groups will help guide doctors to decide which treatment are most suitable, and the breast cancer is divided into the 4 main subtypes that are characterized by the presence or absence of certain receptors on the cell surface. So, cell receptors are proteins that located on cellular membrane, and they act as a lock on the door. So when the ligand binds to it, the internal signaling pathway starts inside the cell. So breast cancer cells are characterized by presence or absence of 3 types of receptors: estrogen receptors, progesterone receptors, and human epidermal growth factor receptor type 2. First group of breast tumor is lumina A, that includes tumors that are estrogen receptor positive and progesterone receptor positive but negative for human epidermal growth factor receptor type 2, meaning that they do not have that receptor on the surface. These cancers are likely to benefit from hormonal therapy and also may benefit from chemotherapy. Second group is Lumina B, that includes tumor that are estrogen receptor positive, progesterone receptor negative, and positive for human epidermal growth factor receptor type 2. The third type is HER2 positive breast cancer. This includes tumors that do not represent estrogen receptors and progesterone receptors on their surface, but they have only human epidermal growth factor receptor type 2. And these cancer are likely to benefit from chemotherapy and the treatment that targets HER2 positive cells. And the last group is triple negative breast cancer, also known as basal-like breast cancer. It includes tumors that do not represent any of those receptors. And this type of cancer is likely to benefit from chemotherapy. So understanding more about the chemical and genetic makeup of cancer will help doctors to choose the most effective treatment for specific cancer, and also it will help researchers to aim the specific cancer types. Current standard therapy combines the precise staging of cancer and prescription of chemotherapy, radiotherapy, hormonal therapy, or surgical resection. And radiotherapy and chemotherapy are well known for their significant side effects, which include fatigue, hair loss, weight loss, problems with memory and sleep. So the main limitation of conventional therapy include non-specific biodistribution. Drugs used in therapy can easily affect tumor cells and healthy cells as well, which result in death of tumor and healthy cells. Therefore, we get the systemic toxicity as other limitation, and side effects arise when treatment affects healthy tissues or organs. Also, they have low therapeutic index, and low therapeutic index means the ratio of blood concentration at which drugs becomes toxic and to the concentration at which the drug becomes effective. So the higher the therapeutic index, the safer is drug. However, most of the medicine that used for chemotherapy, they have low therapeutic index, and that's why patients taking chemotherapy usually under the close control of doctors. Moreover, therapeutic agents are not protected from the degradation within our system, and they have low circulation half-life, and by the time they reach the tumor site, they may lose their therapeutic properties. Most drugs that used are hydrophobic, meaning it's difficult to dissolve them in water, and their poor solubility leads to low pharmacokinetics, so it's difficult to control the absorption and distribution of the drug in the organism. The other challenge of conventional therapy is multi-drug resistance development. As medicaments are taken repeatedly, cancer cells may develop resistance to that medicine, so that is why during the chemotherapy several medications are prescribed, which leads to higher toxicity. Therefore, current treatment procedures in need of targeted delivery systems, which will offer to deliver the molecules to the affected cells in order to help treat the disease without causing any negative effects to the healthy cells. And nanotechnology provides nanoparticles that target chemotherapeutics directly and specifically to cancerous cells, which will result in increased treatment efficiency, increased specific localization. It will also decrease the toxic side effects because we can reduce the dosage, we can control the biodistribution and modulate the pharmacokinetics, and ultimately it will improve patient compliance. So, use of nanotechnology-based therapeutic agents will lead to the decreased risk of the patient and improve survival rate. The use of nanotechnology in cancer treatment will increase the possibility of destroying cancer tissues with minimal damage to healthy tissues and organs. So what are nanoparticles? Nanoparticles are ultra-small particles with size ranging from 1 to 100 nanometers. They have unique physicochemical properties that attract the attention of researchers in medicine, engineering, chemistry, and other areas. As you can see on this scale, the size of nanoparticles is similar to the size of biological molecules and much smaller than the cell. Therefore, they are easily penetrate through the cell membrane and can accumulate inside the cell. Nanoparticles are very promising objects both in cancer diagnosis and treatment, since it can enter the tissue at molecular level. Nanoparticles are now being designed to assist therapeutic agents to pass through biological barriers, to mediate molecular interactions, and to identify molecular changes. They also have large surface area with modifiable optical, electronic, magnetic, and biological properties compared to macromolecules. Nanoparticles can be formulated from any material, and first nanoparticles were made from polymers, which include chitosan and polylactic glycolic acid, or PLGA. Other type of nanoparticles are lipid-based nanocarriers, which include liposomes, etc. In recent years, inorganic nanoparticles are gaining much interest, which includes metal nanocompositions like gold, titanium, silver, and iron nanoparticles, and also silica nanoparticles. Moreover, these nanoparticles may be combined in order to achieve multifunctional properties and can be used simultaneously to provide contrast for different imaging modalities such as MRI, ultrasound scanning, and targetedly deliver of drugs or genes, or used in thermal therapy. Also, viral capsids can be used as nanocarriers, and different conjugates of drugs with different molecules, like antibodies or polymers, can be used in order to protect the drug from degradation and guide the therapeutic agent to the site of action. So, in order to achieve effective drug delivery, two basic requirements should be realized in the design of nanocarriers. First of all, drug should be able to reach the desired tumor sites after administration with minimal loss to their volume and activity in blood circulation. Second, drug should kill only tumor cells without harmful effect to healthy tissues. And these requirements may be enabled using two strategic strategies: passive and active targeting of drugs. So, passive targeting takes advantage of unique pathophysiological characteristics of tumor vessels, enabling nanodrugs to accumulate in tumor tissues. As tumor needs nutrients and oxygen to grow and spread, it forms new blood vessels, and this process is known as angiogenesis. And newly formed vessels are highly disorganized and dilated with high number of pores, resulting in enlarged gap junctions between endothelial cells, like here. And this leaky vascularization refers as the enhanced permeability and retention effect, allows migration of macromolecules up to 200 nanometers in diameter into the tumor microenvironment. However, passive targeting suffers from several limitations. Because universal targeting of cells within a tumor is not always feasible, because some drugs cannot diffuse efficiently, and the random nature of approach makes it difficult to control the process. Moreover, some tumor types do not exhibit an APR effect, and the permeability of vessels may not be the same throughout a single tumor. However, there are drugs that FDA approved and used in clinics. For example, Doxil, it's doxorubicin incorporated in liposomes, and Caelyx, it's doxorubicin encapsulated in liposomes with surface-bounded methoxypolyethylene glycol. And one way to overcome the limitation of passive targeting is to attach affinity ligands such as antibodies, peptides, aptamers, or folic acid or other small molecules that only bind to specific receptors on the cell surface, to the surface of the nanocarriers by a variety of conjugation chemistries. So nanocarriers will recognize and bind to target cells through ligand-receptor interactions by the expression of receptors or epitopes on the cell surface. In order to achieve high specificity, those receptors should be highly expressed on tumor cells, but not in normal cells. And this targeting method is known as an active targeting. The thesis of my research is the development of cell-penetrating silica nanoparticles for drug delivery, and ultimate goal of the project is to formulate nanoparticles that target drugs to cancer cells and prevent adverse effect of drug to healthy cells. And to achieve this goal, two aims should be performed. First is the development of nanoparticle itself that will be non-toxic to human and environment. Also, the formulated nanoparticle should target HER2-positive breast cancer cells. Silica nanoparticles are known for their unique advantages as a delivery carriers, which include excellent biocompatibility, systemic stability, and resistance to pH changes, and also large multifunctionality. So the synthesis of nanoparticles are cheap, easily reproducible, and the size of particles can be controlled during the synthesis. Also, these nanoparticles are biocompatible, biodegradable, and the biodistribution of nanoparticles can be modulated by a different modification of its surface. Also, the release of the drug can be controlled by different external stimuli, including pH difference of healthy and cancerous cells, and various enzymes that are abundant in cancer cells. Also, silica nanoparticles can be used to deliver genes, protein, antimicrobial agents, and drugs. This is the schematic representation of the synthesis of nanoparticle, and at the beginning we have a precursor that acts as an initial molecule to form nanoparticle. And after the following steps, we will get spherical nanoparticles with tile groups on their surface, and the chemical subcomposition of the substance can be analyzed by Raman spectroscopy, which is a chemical analysis technique that provides detailed information about chemical structure of material. On this graph, we are interested in tile groups here, because this group will be used for further modifications of the surface. So via this group, we can link or attach different molecules like antibodies or polyethylene glycol or PEG. That's the water-soluble polymer. So the process of pegylation or attachment of polyethylene glycol onto the surface of nanoparticle is very well known process that protects nanoparticles from detection by the mononuclear phagocyte systems, which increases the blood circulation time and reduces immunogenicity and enhances the therapeutic efficiency of the drug. So after the synthesis, nanoparticles should be further characterized, and here you with results of the three types of nanoparticles. That's a dilated nanoparticle, nanoparticle that were pegylated with 750 and pegylated with PEG 5000. 750 and 5000 denote the molecular weight of the polyethylene glycol, and the molecular weight depends on the number of this group here. So the larger the number of that group, the higher the molecular weight. So we can say that the size of nanoparticles did not change significantly even after the pegylation with heavy PEG. And also the image here is taken on transition electron microscopy, and the magnification 33,000 times. And TM is the microscopy technique in which a beam of electrons is transmitted through a specimen to form projection image. Nanoparticles for biomedical application should be monodisperse, meaning they should contain particles of uniform size. And a PGI here and TM image provide information about the monodispersity, and we can see that the nanoparticles are uniformly. So each type of nanoparticle should be studied for the toxicity before the application on animal subjects. So first we check the influence of nanoparticles in vitro. In vitro means outside the living organism. For that we use cell lines that were derived from different body parts. Here we evaluated toxicity on two cell lines. First is human embryo kidney cells, and there is a breast cancer cell isolated in 1970 from a 69-year-old Caucasian woman. The method that used to evaluate toxicity is known as MTTSA among biologists and used to measure cellular metabolic activity as an indicator of cell viability and proliferation. And this method is based on the reduction of yellow tetrazolium salt to purple formazan crystals by metabolically active cells. According to the International Organization for Standardization norms regarding the evaluation of medical devices, if cell viability decreases by 30%, or if the cell viability falls below 70%, approximately here, then the material should be considered toxic and hence not biocompatible. And our results showed that the nanoparticles didn't cause any toxicity or significant cell death. So as my aim is to internalize the nanoparticles to the cell, I am interested in how it can be achieved. For that we use optical microscopy technique known as confocal laser scanning microscopy, which allows optical slicing throughout the tissue. And we evaluated the permeability of nanoparticles through cellular membrane. On this slide you can observe the HEC293 cell lines incubated for 2, 4 and 6 hours with nanoparticles. Nucleus is represented in blue color, actin filaments or cytoskeleton in red, and nanoparticles in green color. And on these images we can clearly see that nanoparticles penetrate through the cell membrane and accumulate in cytoplasm after 6 hours. However, the pegylation decreased the number of internalized nanoparticles, and this can suggest that pegylation of nanoparticles may redeem nanoparticles from in vivo toxicity, helping to escape from immune cells. On this slide we can see MCF cell lines that were treated with nanoparticles for 2, 4 and 6 hours, and here again we observe that pegylated nanoparticles have lower permeability, and moreover nanoparticles did not disrupt actin filaments of the cell, and also under the bright light we didn't see any cellular changes when compared to control. And here is the 3D representation of HEC293 cell line treated with dilated and pegylated nanoparticles after 6 hours incubation. And red color again is actin, blue color is nucleus, and yellow color nanoparticles. Here we can clearly see that intracellular number of dilated nanoparticles much higher compared to the intracellular number of pegylated nanoparticles, and 3D models on other cell lines showed similar picture. So, in order to target HER2 positive breast cancer cells, we will conjugate antibodies anti-ERB-B2 and CRB2 onto the surface of nanoparticles by click chemistry, and the success of conjugation will be evaluated by enzyme-linked immunoabsorbent assay or ELISA. The steps are represented here. And also you might know about ELISA from the serological test for the detection of total antibodies for COVID-19 that are done at Olymp. So after the successful conjugation of antibodies, we propose that nanoparticles will specifically target HER2 positive breast cancer cells only. So after the collection of data from the experiments in vitro, and if the data shows that there is no toxicity, we can proceed to the next steps, which is in vivo studies or experiments on living organisms. And we should, before applying these nanoparticles to human, we should check whether those nanoparticles are toxic or causing any side effects to living organisms. In our case, it's mice. So we use two mice strains called BALP-C. It's albino lab-bred mice and inbred C57 black mice. And I'm not showing the results from the mice experiment because the content may be sensitive to some of you. So what we did, we injected nanoparticles to the tail vein of mice and observed the biodistribution of nanoparticles. For that we used the imaging system called IV Spectrum. So that captures and detects labeled nanoparticles in living organisms, and results showed that nanoparticles accumulate in liver and spleen 6 hours after the injection, and further investigation is ongoing at the moment, and up to now we didn't observe any phenotypic or behavioral changes on mice. So that's it about my project, and now let's talk about the PhD at Nazarbayev University. So School of Engineering and Digital Science, School of Science and Humanities, and School of Medicine are offering following programs for PhD. And for admission to the program, you should submit the application form, CV, and letter of motivation on the website. And also you should have master's degree or equivalent. Your GPA should not be less than 3.0, and overall IELTS not less than 6.5. But if you graduated from English taught university, you don't need IELTS. GRE test is optional but preferable, and most importantly, you should have very high motivation to study, because during the PhD you will not have fall, spring breaks or summer holidays, so for you there will be only one science or PhD season. So thank you very much for your attention. Hope that lecture was beneficial to you and you got a general idea of what students in biomedical science do. If you have any questions about my research area or the PhD program at NU, please do not hesitate to contact me. And good luck with all your exams. Thank you. Bye.